And next, we have Dr. Lawrence Hayward, who will present an overview of the ongoing Propel clinical trial.
Okay, hello everyone. I'm Larry Hayward. I'm a neuromuscular neurologist at UMass in Worcester, Massachusetts. And I'm new to this field, and it's been just tremendously informative and inspiring and humbling to be here today. I'm going to give you a brief update of our implementation of Propel at UMass so far. These are my disclosures. I'm the principal investigator at our site, and my institution, UMass, received support from Swan Bio for this study. I don't have any other financial link to Swan Bio. And I also work on muscular dystrophy and some unrelated studies. So this is our team that I am just very excited and fortunate to work with. So within neurology, my longstanding colleague and mentor, Bob Brown, who is a world-renowned expert in ALS and also has expertise in gene therapy for that condition, as well as our amazing set of coordinators and our lead research coordinator, Catherine Douthright, who is interacting directly with the patients for screening, as well as our other coordinators. We're also linked up with the Gene Therapy Center at UMass. The dean of our medical school, Dr. Flott, is an international leader in the field of AAV gene therapy. And so we're working closely with his team, as well as the laboratory team at the Gene Therapy Center that have done a lot of the immunological assays and monitoring of our patients. We have a fantastic radiologist who is to deliver this AAV product. We enter the spine at the lumbar level with a small catheter and thread that up to about the T10 level to perform the infusion, which takes about 5 hours, very slowly pumped in. And we have the support from our drug services group for clinical trials as well, which has been fantastic. So as Chris mentioned, and as you all know, in AMN, what we're most concerned about is degeneration of these spinal pathways, either within the spinal cord or the dorsal root ganglia that contribute to a lot of the symptomatology. And so that's what we want to target with this therapy. And as Chris also mentioned, we're using the ABCD1 gene as the cargo. The AAV protein shell encloses the cargo and is the main delivery unit in this treatment. And in this treatment, we will be doing a single injection of a large number of these particles into the fluid surrounding the spinal cord at that thoracic level. So whenever you think about a gene therapy with AAV, you think about how could the body react to this? And one of the main concerns that comes up in various trials is, could there be some sort of immune reaction when you introduce these foreign particles? And both the capsid proteins, the proteins around that shell of AAV, and the protein product that we're introducing, the ALD protein, could be immunogenic. There are cells called antigen presenting cells that recognize foreign objects in the circulation and then signal to what's known as the innate immune response. This is an ancient system of surveillance for foreign infection, other particles, and has its own set of inflammatory mediators and inflammation that we can detect through monitoring. But it's more complicated than even that, because these antigen presenting cells can also activate other cells known as T cells that can, and there are different types of T cells. One type of T cell, if it's presented with antigen, it goes out and looks for any cells that contain that and destroys them. And that's the last thing we really want. We don't want the cells that have taken up our gene product to be killed off. other T cells are, when these CD4 T cells are activated, they can trigger B cells to start making antibodies. These are cells that elaborate antibodies that float around in the circulation and can inactivate our gene therapy product. So we don't want that outcome either. We're learning a lot more about different mechanisms that are built in to sort of to learn to tolerate these antigens. And there's a lot there. These are called T regulatory cells. And so I'm encouraged, there's a lot of research going on to try to harness the properties of those cells to keep these deleterious reactions in check. At the stage we're at right now, what we need to do is monitor patients really closely with blood draws every week or two to detect any sort of the hint of a start of any sort of immune response. Typically in other AAV trials, it's the first 3 months after the treatment that you may start to see some immune reaction, either to the protein product or the viral capsid. And these can be treated by immunosuppressants like prednisone or other agents as needed. And so these are very expected to happen, and we have a way to deal with it. But the key is to be monitoring. And so, as Chris said, we're delivering this into the area of the spine where we will most likely be able to improve the phenotype of the disease. So this trial, the main goals are safety, determining a tolerable dose, and preliminary effects on efficacy. So, and as I mentioned, it's a single administration. The way it has been designed, we started with the lowest dose. We've dosed several patients with that. The plan then is we go to the safety board to get approval to go to the next level. We've done that, and so we're in this second group here that's getting a higher dose. And depending on the results of this, then we may go to higher doses. The main point of this trial is to determine safety, determine what kind of reactions might occur. It's AAV9, which is chosen because it has been used before inside that spinal fluid area, and that shields it from a lot of those antigen-presenting cells that are out in the blood in the circulation. But it doesn't mean that none of that AAV doesn't get out there. And so we're monitoring the liver, for instance, that can be sort of an early warning sign of an immune reaction. And as Chris mentioned, we're really, for this particular study, it includes men age 18 to 65, have to have a genetic diagnosis. And essentially, we want patients that have a mild to moderate degree of severity. We want patients that we can have, we see some phenotype, some problems that we may have some sense of whether things are slowing down or not, because we're going to be monitoring patients over at least 2 to 5 years. So this is a huge commitment on the part of the patients to participate. It involves a number of visits that have to be in person in Massachusetts. The exclusion criteria are we can't accept people in the trial that have evidence of cerebral inflammatory disease. Patients have to be able to tolerate a variety of different immunosuppressants in case they're needed. And diabetes would be a condition that could perhaps be adversely affected by steroids, so we exclude those patients. And patients who may have had a previous AAV would not be eligible either because they build up a response. So essentially the way this is done is our radiologist under fluoroscopy inserts the catheter. We have a pump going. It literally takes about 5 hours, and then we monitor the patient in the hospital for a total of 3 days leading up to this, giving high-dose steroids for those 3 days, then the patients stay in our local area for about a week after the procedure, just to follow and make sure there haven't been any side effects. And the overall schedule then is this screening period to select patients, the treatment, staying around for a week for various other tests. Things that we follow in the in-person visits include a full physical and neurological exam, tests of mobility and balance, imaging, and then collection of blood and other samples to monitor not only for side effects, but also the extent that the virus might have gone out into the rest of the body. The patients also contribute a lot in terms of reporting their global impression, showing us their walking, filling out surveys, and answering other questions. So then the patients go home, and we have a follow-up visit at 1 month, then at 3 months, and thereafter it's about every 3 months for the first year, then every 6 months. So this is at least a 2-year commitment, but we want to follow them for years after that to get some sense of whether their progression has been altered by the treatment. So I'm going to stop there in light of the time, and I'd be happy to take any questions.
Thank you very much, Doctor. In the interest of time, we are going to hold questions. I'm so sorry, guys, but we are still running way behind schedule. So thank you again, Doctor.